It is 2 o'clock and it is time to start the webinar. My name is Hanne Onsted Hansen and I'm a coordinator at the Danish Diabetes Academy. We're delighted that you have joined our webinar with Professor Decio Eizirik. And of course, thank you to Professor Eizirik for accepting to speak at this webinar. Just a few practicalities before the webinar starts, the presentation starts. Professor Eizirik has prepared a presentation of approximately half an hour. Afterwards, we have set aside approximately 15 minutes for questions and discussion. If you have a question or comment, please use the chat function. You open the chat function by clicking at the chat icon at the bottom right corner of your screen. You can ask your question in two ways. You can either simply write your question, or if you prefer to ask your question verbally, write "I have a question" or something similar, then I will unmute you. Please do not ask to be unmuted if you have background noise, as background noise is very disturbing. And it will be very helpful when you write your questions that you write them to all participants or to everyone. That was all from me. I will stop sharing my slides, and I hope you all will enjoy the webinar. So, Professor Eizirik, should be able to share your presentation now.
Yes.
Perfect. Yes.
Okay. Well, it's a pleasure for me to talk to you today, and we are in an interesting situation because I am a visiting professor who is not allowed to visit thanks to the COVID. So anyway, we do the best of the situation. So we'll have this webinar today, and I will be happy to discuss further with those of you who wish to ask me questions. So what I'm going to discuss today is the dialogue between target tissues and the immune system in type 1 diabetes mellitus and other autoimmune diseases. And the data that I'm going to present today was generated in my two labs, the first one in Brussels and the other in Indianapolis. And the main people involved are Michael Colley, Florian, Maria Inês Alvelos, and from the Indianapolis side, Stefan Denis. And I have several collaborators that I'm going to mention during the presentation. In Indianapolis, we have a very close collaboration with Carmela and Raghu, and several other colleagues that I'm going to mention. And of particular relevance is in the context of this DDA visiting professor fellowship is Fleming Possio, who is my host at the Destino Diabetes Center and with whom I have been collaborating for many years. So, as most of you know, type 1 diabetes is a chronic autoimmune disease where you have individuals at risk for the disease. At some stage, something happened that we don't know still what it is. It may be, in some cases, a viral infection. In others, this remains an open question. And this will lead to what we call stage 1, which is when the individual developed two autoantibodies against beta cell antigens. And people who have two autoantibodies, they have more than 80% chance to develop diabetes in the coming years. So since this is an autoimmune disease, most of the focus in the field has been in the immune system. And we and other colleagues have been proposing a slightly different view, that this is not a disease only of the immune system, but is a disease of a misguided dialogue between the immune system and the pancreatic beta cells. What triggers this misguided dialogue remains to be clarified. It may be, again, a viral infection, and pancreatic beta cells has a quite well-defined gene network to recognize and to respond to viral infections. It can be a non-specific inflammatory triggering, leading to cytokine production and cytokine receptor signaling. And this will lead to a next step, which is activation of key transcription factors, including, for instance, STAT1 and STAT2 and nuclear factor kappa B. And it's interesting that some candidate genes for type 1, like ETP and 2, they act here down-regulating activation of STAT1 and STAT2. The next stage, let's say, in this response is what we call the effector phase, where you will have both upregulation of MHC class 1, alternative splicing, and endoplasmic reticulum stress, which may lead to presentation of modifier antigens in the context of both MHC class 1, and if the dying beta cells are phagocyted by dendritic cells in the context of MHC class 2. There will be also pro-apoptotic signaling, and the beta cells produce chemokine, like for instance CXCL10, that will attract additional immune cells. What's to keep in mind is that not all signals triggered by cytokines are deleterious. For instance, as I'm going to show, beta cells exposed to pro-inflammatory cytokines, they express PD-L1 or HLA-E, which are inhibitory molecules for CD8 T cells. So one of the reasons that led us to propose this hypothesis that beta cells are not inert victims of the immune system, but they are active participants in the disease, is that when we look at the candidate genes for type 1, and there are more than 50 regions today, people usually assume they are all related to the immune system. However, when we cross these candidate regions against the RNA sequencing that we have done of human islets of Langerhans, what we found is that more than 80% of the candidate genes for type 1 are present in pancreatic islet cells. Since we have so many genes, this allows us to look at which are the pathways that these candidate genes regulate. And it's interesting that they regulate recognition of a viral infection, for instance, via MDA5, and several of the candidate genes are involved in the signaling of type 1 interferon, starting with a kinase TYK2, which is crucial for signaling of the type 1 interferon receptor. And it's interesting that polymorphisms that decrease but not abolish TYK2 expression are protective against type 1. And then you have several other candidate genes, two of them, GLIS3 and CTSH, which is something that Fleming and colleagues have been studying, are more related to the beta cell function. But so this points to the relevance of type 1 interferon in the disease process. And indeed, type 1 interferon is a key cytokine in the early stages of type 1, and is the cytokine that is most consistently found when you do histology of the islets of patients with type 1. And interferon is also a key player in the dialogue between pancreatic beta cells and the immune system. And its role is via induction of inflammation, HLA class 1 expression, and endoplasmic reticulum stress. And these three are hallmarks of beta cells in type 1. And if you look at the way type 1 interferon signaling is via binding to a surface receptor. It will just then activate two kinases, JAK1 and TYK2, and as I told you, TYK2 is a candidate gene for type 1. These two kinases will phosphorylate two transcription factors, STAT1 and STAT2, that will form dimers and then bind to IRF9 or IRF7 and go to the nucleus where they will stimulate interferon alpha beta stimulated genes. So to better characterize the effects of type 1 interferon on human beta cells, we did a multi-omics time course analysis, where we, in this case, treated either endo beta 1 cells, it's a human beta cell line, or human islet of Langerhans with interferon for 2, 8, or 24 hours, did RNA-seq and attack-seq, which allow you to see areas of opened chromatin, and did in parallel, at least at 8 and 24 hours, proteomics, and then data integration. One thing that is important when you are using this in vitro model is to see if they indeed recapitulate the gene signatures that are present in patients with type 1. And this turned to be correct, so we were fortunate to get access to databases of RNA sequencing of FACS-purified beta cells from 4 type 1 patients as compared to 12 controls. So we compared this data set against our beta cells, human beta cells treated with type 1 interferon, and you see that there is a very good concordance related to upregulated genes and no concordance on downregulated genes. This is not a non-specific effect of beta cell stress, because when we look at islets from type 2 patients and compare with interferon treatment, there is no similarity. And we also compared beta cells from type 1 patients against islets, human islets treated with interleukin-1 beta plus interferon gamma, and there was again a fairly good correlation, and again a minimal or no correlation with type 2. So when we start to look at the data, the first thing that called our attention when we look at attack-seq is that there is a very early effect of type 1 interferon. It's a massive opening of the chromatin after 2 hours, when this, let's say, this chromatin opening is reduced after 24 hours. And this very early response makes sense because type 1 interferons, one of their functions is the antiviral response. And since virus proliferate very, very fast, it is crucial the cellular response is also fast. So when we integrated the RNA sequencing and the proteomics, we could identify several relevant patterns, including, of course, interferon signaling, endoplasmic reticulum stress, beta cell deaths, and so on. One thing that was interesting is that we found regulatory mechanisms, which is in line with this concept that not only signals generated, not all signals generated by interferon will be deleterious to the beta cells. Some of them are involved in beta cell protection. So we went a little bit further, and in a previous study that we published last year that we did together with Lorenzo Pasquale, we have also looked not only at attack-seq proteomics and RNA sequencing, but also at SHIP-seq, which is a way to look at areas of the chromatin where transcription factor is indeed binding to the neighbor, the DNA area. And when we started to look, let's say, in both beta cells, human beta cells, all the data that I am showing today is human. So this presentation at least today, is a mouse-free presentation. So when we treated human beta cells with interleukin-1 beta plus interferon gamma, or a type 1 interferon, you will see bindings of transcription factors that you would expect, like IRF1, STAT1, STAT2, and so on. But to our surprise, we also found binding of several transcription factors that are involved in maintenance of the beta cell phenotype, like, for instance, PDX1 and X61 NeuroD. And when we did a correlation analysis to try to see if they were binding to the same regions, again, to our surprise, we found that, for instance, PDX1 was bound to several genes together with ISRE, which is a classical binding site for type 1 interferon-induced transcription factors. There was also a clear correlation with STAT. So based on this, we developed a concept suggesting that most probably these binding sites that are bound by transcription factors that are involved in the beta cell phenotype, they open the chromatin and they facilitate binding of transcription factors induced by the inflammatory process. And another thing that was interesting in this and in a subsequent study, when we compared, let's say, the binding sites of candidate genes for type 1 and type 2, what we find is that in the case of type 1, the binding sites are mostly in islet stable regulatory elements. So they are bound under basal condition. So if you do EQTL, you are able to detect them. And which are the genes, let's say, where these candidate genes are actually binding? Well, they are involved in insulin secretion, glucose sensitivity, and beta cell identity, which makes sense in the context of type 2. When you look in the context of type 1, you do not see binding under basal condition. You will only see binding after the islets are exposed to an inflammatory stress, in this case, cytokines. So this will help transcription factors to bind. And in this case, they are binding, let's say, to regions regulating apoptosis, immune response, and beta cell stress. So this suggests that if you want to do EQTL for candidate genes for type 1, most probably you cannot do it in the basal condition. You need to do with stimulated beta cells. At that stage, I was starting to be curious because we have always been studying type 1 diabetes mellitus and suggested that in this particular autoimmune disease, there is a dialogue between beta cells and the immune system. And I started to wonder if this would be a pure type 1 phenomenon, or it would be also true in other autoimmune diseases like lupus, multiple sclerosis, and rheumatoid arthritis. And now there are, in the literature, and in particular databases like GEO, you have data sets from target tissues of these diseases that you can download, process in your own pipeline, and study in a comparative way. So for the pancreatic beta cells, we had 4 type 1 and 12 controls. For lupus, these were kidney cells with lupus nephritis. There were 20 patients and 7 controls. For multiple sclerosis, it was optical chiasm, 5 patients and 5 controls. And for rheumatoid arthritis, where it's much easier to make biopsies, there were a much larger number of patients and controls, 57 and 28. So, as I told you, we were able to retrieve all the raw RNA-seq data. We quantified it using a program called Salmo, did the differential expression analysis using another tool, and then we divide it, let's say, in two analysis pathways. One is pathway enrichment analysis, and the other that I'm going to show to you in detail is rank-rank hypergeometric overlap, which was followed by additional analysis. So when you do pathway enrichment analysis, and here you have the four diseases, type 1, lupus, multiple sclerosis, and rheumatoid arthritis, you see that there are several pathways that are upregulated, and many of these pathways are upregulated in common. For instance, antigen process or presentation is present in all target tissues of these autoimmune diseases. However, when you look at the downregulated genes, you have a different picture. In the downregulated genes, they tend to be disease-specific. So, for instance, in the case of type 1, the most downregulated pathway is regulation of gene expression in beta cells. When you are looking at multiple sclerosis, you have protein-protein interaction at synapses. When you are looking at rheumatoid arthritis, you find, for instance, collagen chain trimerization. So it seems that the downregulated genes are disease-specific, while the upregulated genes tend to be similar, to the extent that the highest upregulated pathway in type 1 is lupus. So there are so many genes in common that lupus comes as the most upregulated pathway. And the other thing that that was striking is that in nearly all diseases, we were finding interferon signal, both interferon gamma and type 1 interferon. Sometimes they are not in the top 20, but they were always present. And when we are studying these cells, you always have a very minor immune cell contamination. So to give you an idea, we quantified CD55-4, which is a marker of immune cells, and the transcripts per million in type 1 was around 16, while the transcripts for insulin are more than 100,000, and for PDX1 around 90. So it's a minor contamination, but to exclude that what we were seeing was not due to the immune cell contamination, we redid the analysis after downloading data sets where people study single cell RNA. And again, in type 1, we got a few beta cells, and in all these autoimmune diseases, we could find a clear and significant interferon-stimulated score. So this upregulation of interferon in the target tissues is real and cannot be explained by the immune contamination. We then went on to compare diseases, and we used this tool, rank-rank hypergeometric overlap. And in this type of tool, you are not using a cutoff. You just rank, let's say, genes, compare type 1 against control. And in multiple sclerosis, we compare optic chiasm of patients with multiple sclerosis against controls. And here you have genes that are upregulated in both diseases, and the intensity of the color gives an idea of the degree of significance. So there are a very large number of genes upregulated in common at an extremely high significance level, and there is very few downregulated genes in common. When you put all diseases together, so here you have the image. So for instance, here you have systemic lupus compared to type 1, and you see that you have 624 genes in common. Here you have something interesting. You have nearly more than 700 genes that are upregulated in type 1 and downregulated in lupus nephritis. So these diseases, in this case, have also some marked difference. In the case of multiple sclerosis and rheumatoid arthritis, as compared to pancreatic beta cells, they have mostly upregulated genes in common. As I told you before, when we look at the expression of candidate genes for type 1 at the beta cell level, we had seen in our human islet preparation in vitro around 80% of the candidate genes present. And now looking at RNA-seq of beta cells from patients and controls, we find again 80% of the candidate genes expressed. Is this a pure beta cell type 1 phenomenon? No, because when we look at lupus, multiple sclerosis, and rheumatoid arthritis, we find between 83 and 92% of the candidate genes present. Again, we wanted to exclude that this was due to immune cell infiltration. So we went to the single cell RNA-seq, and you find, let's say, in all of these target tissues, expression of these candidate genes. One thing to keep in mind is that when we are doing bulk RNA-seq, we can have a very deep depth of sequencing. We sequence at 200 million. When you are doing when you do bulk cell RNA-seq, we can detect around 50,000 transcripts because we are getting both whole genes and splice variants. When you are doing single cell RNA-seq, you are getting 2,000 to 3,000 genes. So in this case, at the single cell level, we look at the top 50 candidate genes. And one interesting candidate gene that you always find in all diseases is TYK2. So it's present in the beta cells, is present in kidney epithelial cells of patients with lupus, is present in neurons, and is present in synovial fibroblasts. So this, as you probably remember, is the kinase involved in type 1 interferon signaling. And indeed, when we look at the risk genes, let's say, putting together the 4 diseases, you find that TYK2 is one of the 4 candidate genes in common between the 4 autoimmune diseases. You have other genes that are disease-specific, many that are present in 2 or 3 diseases. So we grow very interested in TYK2, and why? Because when we look, let's say we can mine our data sets for drug discovery, and basically what you try to do is compare your own data sets against available data sets in the Broad Institute, where they treat the different cell lines with different drugs. So we try to find drugs that antagonize our gene and protein signatures. And one of the clear-cut ones was JAK1, who antagonize, are drugs that block JAK1, which would inhibit STAT1. And indeed, we have looked at one of these drugs, baricitinib, that is already used to treat rheumatoid arthritis, and we could completely prevent interferon alpha induced HLA expression, and so on. However, we were more interested not only in JAK1, but particularly in TYK2. And why TYK2 is an interesting candidate for therapy? Because there are several studies involved, let's say, in drug development, showing that drug development programs supported by human genetic data are more likely to be successful. So after our initial data with JAK1 inhibitors, we went to TYK2 inhibitors. And to make a long story short, what we found is that TYK2 inhibitors that are already being used, for instance, to treat psoriasis, they completely block interferon-induced phospho-STAT1 and phospho-STAT2, and this is quantified here. They prevent HLA class 1 expression. And here you can see a histological picture. So you see that when you expose human islets of Langerhans to interferon alpha, you have a major increase in MHC class 1. It's mostly at the beta cell level. And this is prevented by TYK2 inhibitors. These TYK2 inhibitors also prevent expression of a chemokine, CXCL10, and prevented endoplasmic reticulum stress. So the conclusions and future plans, and all that you see in blue here are future plans as part of my visiting professorship, with eventually I am allowed to visit in collaboration with Fleming, Possio, and colleagues. So our data suggested human islets obtained from individuals affected by type 1, but not type 2 diabetes mellitus, present a gene and protein signature that is similar to that induced by in vitro exposure to interleukin-1 beta plus interferon gamma or interferon alpha. Interferon alpha promotes early changes in chromatin accessibility that are probably necessary for a rapid response during a viral infection. Exposure of human beta cells to type 1 interferons or to interleukin-1 beta plus interferon gamma activates several cis regulatory elements, many of them presenting enrichment for islet-specific transcription factor and impacting on expression of candidate genes for type 1. These primed regulatory elements may facilitate cell type-specific responses to pro-inflammatory signals, potentially explaining tissue-specific genetic susceptibility in autoimmune diseases. The second conclusion are that there are major common changes in gene expression at the target tissue level in 4 autoimmune diseases, mainly downstream of type 1 and 2 interferons. 80% of the candidate genes for the 4 autoimmune diseases studies are expressed in their target tissues, suggesting that our previous findings in type 1 are probably valid for other autoimmune diseases. The ultimate function of these candidate genes remain to be clarified, and this is one of the research lines that we are pursuing together with Fleming and his colleagues. And we suggest that target tissues should be considered when studying autoimmune diseases to better understand the genetics and natural history of these serious diseases. And testing the perturbagens that detected in the present study may suggest drugs that can be repurposed for the treatment of different autoimmune diseases. And the first drug that we are suggesting for repurposing is inhibitors of TYK2 that could be used as a therapy for type 1. This is what I would like to show to you today, and I will be happy to answer questions. I just look at the chat and there is nobody chatting. So
No.
if you have questions.
No, not yet. Thank you for your presentation. And as Professor Eizirik said, you now have the opportunity to ask questions to the presentation, or maybe you have a comment. You can write your questions in the chat function. And if you prefer to ask your question verbally, you can just write, I have a question or something similar, then I will unmute you and you can ask your question. I know from previous webinars that sometimes it takes some minutes before the first question comes in the chat because people need time to write them. So, okay, here's the first question. Can you see it, Decio?
Yes, but there is one question. Patrick is...
Yes, okay. Patrick wants to... maybe we can take the question from Magdalena, and then I will unmute Patrick afterwards.
Okay, so Magdalena is asking, well, she's thanking me for this interesting talk. Thank you very much. Have you looked at human islets from patients with LADA? Would you expect similar results as in type 1? Despite the fact that clinically the disease resembles type 2? Unfortunately, we did not look at RNA-seq of beta cells from patients with LADA because we do not have at that moment access to this type of patients. So it's very difficult to predict what we were going to find. There are histological studies from Sarah Richardson and colleagues, and it's not specifically at LADA, but it's looking at patients where type 1 starts at different ages. And they find, for instance, that younger patients, if I'm not wrong, below 13, have a more aggressive disease with a high infiltration of B cells. So this is interesting, and I think this is something that we need to study further.
Yes. I will unmute Patrick, hopefully. Okay. Patrick McDonald, you are unmuted and are now able to pose your question.
Thank you. Can you hear me?
We can hear you, yes.
Decio, I sympathize with your inability to travel to Denmark. I would like to be there also. And I couldn't figure out how to type my chat to everyone, so I thought I would just ask verbally. So my question, I'm very intrigued about the differences in gene expression you see with type 1 islets, type 2 islets, and treatment with interferon and other cytokines. And, I'm thinking about this idea that inflammation, has been implicated in type 2 diabetes and islet dysfunction as well. So maybe, can you comment on between the type 1 and type 2? I think you mentioned it in your conclusion a little bit, but I mean, do you expect, for example, so what's the difference in the inflammatory response in the type 1 and type 2? And do you expect the type 2 islets to become more similar to type 1, say, in very long duration diabetes, when you may have apoptosis, for example?
Well, the problem is that, in my view, based on the data that we have, the role of inflammation in type 2 is very limited. And attempts to treat type 2 using anti-inflammatory molecules or interleukin receptor antagonists have not been successful. So when we compare the gene expression, for instance, of interleukin-1 beta treated human beta cells of islets against islets from type 2, there is a very, very mild correlation. It's like that in type 1 is around 100 and in type 2 is 1 or 2. So we just do not see this supposed interleukin-induced signature. With interferon alpha is 0, you see absolutely nothing. We have published a follow-up paper where you look at the transcripts instead of whole genes, and it's the same picture. So I am not really sure what is the role of inflammation at the beta cell level in type 2. I think for me, this remains an open question.
Yeah, I know, of course, the interleukin story has been a bit controversial, for sure.
Patrick, since you are online, we are very eager to increase the number of our beta cells panel from type 1 patients. So if you have more single cell analysis, we would be very happy to add to the analysis that we have been doing. And
sure, we can talk about this, of course, yeah.
Keep us in mind as you take additional data.
Okay.
So, there are two additional questions from Christian. So again, he's thanking me, and I thank him for listening. So he asks two relevant questions. Does TYK2 inhibition completely reverse the changes or a selective subset? How about protective genes such as PD-L1, HLA-E, and so on? Unfortunately, when you block TYK2, you also block PD-L1 and HLA-E. So you do not dissociate the protective from the deleterious genes. What we are trying to do now is to find ways to dissociate these pathways. And I think this is possible because when you knock down STAT2 in human beta cells, you have a paradoxical increase in PD-L1, while HLA class 1 and chemokines go down dramatically. So we are just screening for drugs that may be able to do this. One thing that I didn't mention, because the other concern that I have with TYK2 and JAK1 inhibitors is that besides inhibiting PD-L1 and HLA-E, they could also prevent the beta cell ability to defend against viral infections. So we also infected these human beta cells with Coxsackie virus, with or without a TYK2 inhibitor, and they responded in the same way. So there was no more severe viral proliferation or more beta cell death. So it seems that if you block TYK2 or JAK1, not completely, but around 50-60%, they keep the ability to respond to virus. Then I have another question, which is also an interesting one and a burning issue. How about COVID-19 diabetic patients? Here is a viral infection and a cytokine storm. Any comment? At this moment, comments are very speculative. So there are case reports of patients who developed type 1 immediately following or in the course of a COVID infection. And there is a paper that is impressed by Francesco Dotta, Guido Sebastiani and colleagues, we had the chance to collaborate, which shows that human beta cells express the receptor for the COVID-19 virus. And this has been also shown in another study based on iPS-derived beta cells. There are two American studies deposited online, not yet peer-reviewed, claiming that this receptor is not there. But a couple of days ago, I saw deposited another paper by colleagues from whom showing again that they have receptors for the virus, that they can be infected, and they went so far as to do RNA-seq, and they have a beautiful, I will call beautiful, they have a clear-cut interferon signature. So there is a possibility that the SARS-CoV virus infect beta cells. I don't think that this will lead to an acute beta cell killing, but this is speculative. My concern, and I hope that I am wrong, is that this type of infection in genetically predisposed individuals may lead eventually to autoantibodies and type 1. But this we'll only know by doing follow-up studies. And then there is a question from my dear friend, Amy, saying that, well, again, congratulating me, thank you. What is your take on the observation that is mainly down-regulated pathways that are specific? That's an interesting question. I think when we are looking at the upregulated genes, we are looking at inflammation-induced genes, and it turned to be that type 1 and type 2 interferon are common mediators. So they are acting on these different autoimmune diseases, and they will lead to upregulation of, let's say, groups of genes that are in common. When you are looking at the downregulated genes, you are looking more at the impact of the inflammation on the target tissue. So, for instance, in the case of beta cells, they are losing their phenotype, they are losing oxidative metabolism, and so on. When you look at multiple sclerosis, synaptic function is being affected. When you look at rheumatoid arthritis, you find defects in collagen. So the impression that I have, I think this needs to be studied further, is that when you are looking at downregulating phenomena, you are looking at the consequences of the inflammation on specific tissues. And this, by definition, would be more tissue specific.
Thank you for answering the question. So, any more questions? No. Then